Hello, this video is going to be dedicated to the process of glycolysis. And if you've been studying this material already, you're going to notice that this is always the first biochemical pathway introduced when you start discussing the process of ATP production. The human body is going to utilize glucose as an energy source. Later on, when you do lipids, you'll note that we'll use lipids as an energy store. The beginning of that process of converting these carbohydrate molecules into ATP is going to be the chemical reactions of glycolysis, where we take one molecule of glucose and break it down into two molecules of pyruvate. One of the important things to notice about glycolysis that's even outside of its reactions is where in the cell it takes place. Glycolysis will always take place in the cytosol of a cell, the liquid component. Most of these biochemical pathways for ATP production will take place in the mitochondria. Hence that tagline that you always see with the mitochondria as being the powerhouse of the cell. Most of our ATP will be produced inside the mitochondria. Glycolysis, however, takes place outside of the mitochondria in the cytosol. Glucose is a 6-carbon ring structure. Whenever you see a diagram like so, all of these points will always represent a carbon molecule. So 1, 2, 3, 4, 5, 6. 6 carbons making up this ring-like glucose molecule. As we take that glucose molecule and we put it through a cascade of reactions, very often it isn't meant to be memorized as far as this whole chain goes for introductory physiology. But there are some key points to pick out so you understand what's been going on. When you see a process of glycolysis, you'll see initially we enter ATP to start the process going. Then we have a split, and we have 2 separate pathways after that. From there, we wind up producing 4 molecules of ATP and 2 molecules of NADH. Our end product are 2 molecules of pyruvate, 2 3-carbon molecules, which should make sense because our original molecule of glucose had 6 carbons. We cut it in half, we should expect to see 2 3-carbon molecules. So for my students, I tend to like to break it down like so. This is what's most important to the people that I show this to. Our starting point is going to be our glucose molecule. We're then going to use the enzyme hexokinase to convert it into a molecule called glucose 6-phosphate. I like to isolate that particular intermediate because it's important as a launching pad for other reactions, particularly the production of glycogen. We can link these molecules together at times when we have excess glucose and store that energy in the liver as a large linked together glycogen molecule. At times when our blood sugar gets too low, we can tap into that resource and take that glycogen and break it down into individual glucose 6-phosphate molecules in order to carry on with the rest of our glycolysis pathway. So once we get to this point, we can store more glycogen or we can continue on our breakdown process. If we continue on, like mentioned before, 2 individual molecules of pyruvate. Here's a little bit of a closer look at our glycogen production with the particular enzymes taking place in each. Notice that this portion of this reaction pathway is the same as our initial step in glycolysis. Glucose getting broken down to glucose 6-phosphate using the enzyme hexokinase. And then we withdraw and deposit as needed to enter into that glycolysis pathway again. When we look at pyruvate, a little distinction that's important to make to keep us from getting confused is that even my students will often hear me interchange pyruvate with pyruvic acid. Those are essentially the same molecule depending on the state that it happens to be in. Here is what the last pathway had indicated as was our product of glycolysis. However, if we add this proton back on for an ionic bond, we will get pyruvic acid. So for our intents and purposes, whichever one gets mentioned, we're talking about the same molecule. So once we get through glycolysis and we've broken down that glucose into 2 molecules of pyruvate, we produced a net gain of 2 ATPs and we produced 2 NADH molecules. We need to kind of envision where do we go from here. We see an average of 36 molecules of ATP produced for every molecule of glucose. When all is said and done, 36. With our glycolysis pathway, as I just mentioned, we got 2 molecules of ATP and 2 molecules of NADH. So we are a far cry away from achieving the full potential that that initial glucose molecule had. We expect to get 36. So far, we've got 2. We've got 34 left to gain in some way, shape, or form. So when we look at glycolysis or any of these other biochemical pathways, such as Krebs cycle, such as oxidative phosphorylation and beta oxidation, you may study them as individual biochemical pathways. But I want you to envision them as individual cogs in a much larger machine of ATP production. They function with each other and they run together with a great degree of continuity. So when we leave glycolysis, the pyruvate that we had produced at the end, as well as our NADH molecules, are going to be transported into the mitochondria. From there, 2 things are going to be observed. We are going to convert our pyruvate into acetyl-CoA. Acetyl-CoA is going to be considered a starting point for our next biochemical pathway, Krebs cycle. So I encourage you to hit the link down below and check that video out specifically as a continuation. Additionally, the NADH that was produced from glycolysis will enter another pathway called oxidative phosphorylation, sometimes called the electron transport chain. Again, click the link down below and check out that individual pathway as well. But generally speaking, what we're looking to do is go from glycolysis in the cytosol, enter the mitochondria, run through Krebs cycle, take all the products that are left behind and put those through the oxidative phosphorylation process to give us the rest of our ATPs to get an ultimate gain of 36. I want to thank you for watching. Again, check out the next video on Krebs cycle for the continuation of this process.